Thank you for the introduction.
Welcome, Raquel.
Can you hear me?
Yes.
Okay, perfect. And thank you so much, Ira, for the kind invitation. It's my pleasure to be here today. So today I would like to take the opportunity to talk about an area which was disregarded in the past, but which is currently gaining attention because of its clinical relevance. This area is the so-called macro-metastasis organ parenchyma interface. Can you see my pointer? Now, okay, or shortly MMPI. So these are my disclosures. Our group is interested in the study of brain metastases. Brain metastases are the most common form of brain neoplasms in adults and the main cause of cancer-related death. So the main cancer types that frequently metastasize to the brain are lung, melanoma, breast cancer, renal cancer, and colorectal cancer, with an incidence of 40% for lung cancer, 50% for melanoma and breast cancer, and 9% for renal and colorectal cancer. Interestingly, recent postmortem studies have shown that the incidence of brain metastasis is even much higher, reaching up to 90% in the case of patients with melanoma. Moreover, brain metastasis confer dismal prognosis with a median survival of less than 1 year. So all in all, brain metastases represent the biggest clinical challenge among cancer metastases up to date. In the era of next generation sequencing, many efforts have been done to try to understand the molecular mechanisms of brain metastases. However, nobody cared about how they look like. This is in part due to the historical assumption that brain metastases are sharply demarcated lesions that grow by just pushing the adjacent brain parenchyma without infiltrate benefits. However, this assumption was challenged by the observation that the local recurrent rates after brain metastasis resection were much higher in patients that only received surgery than in those who received surgery and radiotherapy, suggesting the presence of residual metastatic cells in this area, what we called the macro-metastasis organ parenchyma interface, or shortly MMPI. So recent histological studies supported the assumption that not all brain metastases grow just by displacing the adjacent tissue, like in this case, where we can see a clear separation between the metastatic cells and the brain tissue. In other cases, brain metastases grow by infiltrating the brain, like in this case, where metastatic cells can be found far away from the metastatic core. And most importantly, metastatic infiltration at the MMPI correlated with a worse prognosis in a small retrospective study including 39 patients with different primary tumor types. Interestingly, while 43.5% of patients with displacing metastases survived 2 years or longer, only 6.6% of patients with infiltrating brain metastases overcame this time. And moreover, all long-term survivors belonging to the displacing group, and this included one patient with non-small cellular lung cancer who survived even longer than 4 years. The univariate analysis of this retrospective study revealed that metastatic infiltration was prognostic relevant with a p-value of 0.009 and a hazard ratio of 3.3, meaning that the survival of patients with an infiltrating MMPI was 3 times shorter than the survival of patients with non-infiltrating MMPI. Interestingly, other prognostic factors like the number of metastases and the performance status, or KPS, which are usually significant in larger studies, didn't reach the prognostic significance in our basket trial, indicating that the metastatic infiltration must be even more relevant in studies including more patients. So that means that metastatic infiltration at the MMPI is clinically and prognostically relevant for brain metastasis. And these differences at the MMPI are not exclusive of the brain. In the liver, metastases can also grow either by displacing the adjacent tissue, forming also in some cases a desmoplastic capsule, or by infiltrating it, as it is shown for this patient here. Unlike brain metastasis, the infiltration of liver metastasis routinely assessed in pathological reports. And in this case, they don't refer to it as MMPI as we do, but as histopathological growth pattern, or HDP. And interestingly, also in this study, melanoma patients with infiltrating, or as they call it, replacement HDP, had a significantly worse prognosis than patients with a desmoplastic or displacing MMPI. And in fact, the multivariable analysis of this study revealed that any kind of replacement or infiltration was associated with an almost 4 times worse survival. And also, again, in this study, other prognostic factors like incomplete resection didn't reach prognostic significance, indicating that metastatic infiltration must be much more important in larger studies. So again, infiltration at the MMPI is clinically and prognostically relevant also for liver metastasis. So in the last years, we have been able to identify at least 9 different MMPI patterns in brain, liver, and lung metastasis. We divided these MMPIs into 2 big categories, displacing or infiltrative, depending on whether the borders of the metastasis are well demarcated or not. Displacing metastasis can present without or with a reactive pseudocapsule that demarcates tumor cells from the adjacent parenchyma. Here we can see a very nice example of displacing MMPI without capsule in humans. In this case, it is a patient with lung cancer, brain metastasis, but we also see this pattern in other tumor entities, especially breast and colon cancer. And as you can see here, the border between the brain tissue and the metastasis is clearly defined. And metastatic cells grow only by compression, pushing the normal tissue aside without inducing an obvious reaction of the adjacent organ parenchyma. Apart from displacing metastasis, we have also identified metastases that infiltrate the adjacent tissue. We have 2 different groups, epithelial or diffuse infiltrating MMPIs. In epithelial MMPI, cells infiltrate the tissue as a collective or groups of cells, while in diffuse MMPIs, we observe single cells. Here, I'm showing you an example of a glandular infiltrative MMPI. In this case, a patient with breast cancer, brain metastasis, and as already explained, the borders of epithelial infiltrating MMPIs are not well defined, and these glandular cohorts here are physically separated from the metastasis. And last, here is an example of a malignant melanoma brain metastasis showing diffuse infiltrative MMPIs with single cells infiltrating the brain. So until now, I have shown you that, first of all, brain metastasis can grow as displacing or infiltrative, that metastatic infiltration indeed correlates with a worse prognosis, and also that there is a wide spectrum of MMPIs. So what we asked was, are different molecular mechanisms responsible for the different MMPIs? And in that case, can we identify therapeutic targets for each of the MMPIs? And in order to answer this question, what we did was to develop mouse models of brain metastasis. Our model, as you can see here, consists of the stereotaxic intracortical injection of murine tumor cells into a syngeneic mice, and we let them grow until the mouse develop brain metastasis, and then we can have a look at the MMPI at this area here. So we have currently more than 10 mouse models of breast, colon, and melanoma brain metastasis that resemble almost all MMPIs that we have identified in humans. With these models, we are trying now to understand the molecular mechanisms that are responsible for its MMPI. And this is exactly what we did in this study I'm presenting today. We compared these 2 models of breast cancer brain metastasis with an epithelial MMPI. on the one hand, the 410.4, which infiltrates the adjacent tissue as strands. And on the other hand, the 41 that also cause epithelial infiltrating brain metastasis, but in this case, infiltrates as cohorts of cells. The interesting thing about these two models is that the 41 is a TO1 resistant variant of the parental 410.4. And although both models have the same origin, they show a very different colonization potential. When we inject these tumor cell lines into syngeneic mice, the 410.4 needs 3 times longer than the daughter cell line 41 to develop macro-metastasis. So we wanted to know why. So first of all, we had a look at both cell lines in culture, and we observed that although both cell lines grew adherent and displayed an epithelial morphology, the 41 were more loosely packed than the 410.4 that grew forming tight clusters. Moreover, we saw that while both cell lines expressed similar proteins levels of the intermediate filament cytokeratin 8, they had much lower levels of the epithelial markers ecadherin and beta-catenin, and they expressed higher levels of the mesenchymal marker pimentin. And finally, the 41 had a significantly higher invasion potential than the parental and less aggressive 410.4. So all in all, it seems that the better colonizing 41 cells displayed mesenchymal features, suggesting that the higher colonization potential of these cells might be associated with the induction of the so-called epithelial to mesenchymal transition. As you probably already know, tumor cells at the primary sites usually undergo this EMT process, which consists of the downregulation of epithelial markers like ecadherin and upregulation of mesenchymal features to invade into the adjacent tissue and get into the circulation. And once at the target organ, disseminated cancer cells reverse this phenotype into an epithelial one to form metastasis. However, the next step, how macro-metastases grow, and especially how metastatic cells infiltrate the adjacent parenchyma, has not been looked into detail until now. And at first sight, it seems that the invasion at the primary site and the infiltration at the metastatic site might follow the same rules. So that's why we thought that EMT induction by the 41 might be responsible for their better colonization potential in the brain. So we decided to study the role of the EMT transcription factor LEF1 in our models. And why LEF1? LEF1 plays a very important role during EMT induction. As you can see here, when Wnt signaling is activated, beta-catenin accumulates and translocates to the nucleus, where it binds to the TCF left transcription factors and activates the transcription of some EMT target genes like fibronectin, snail, or twist. Moreover, we knew from the literature that LEF1 influences metastatic colonization in lung cancer and the survival of patients with brain metastasis. And also that LEF1 is commonly overexpressed in different brain colonization breast cancer cells. So first of all, we confirmed that LEF1 was higher expressed in the better colonizing 41, both at the protein and also at the RNA level. However, the expression of AXIN2, the typical target gene of Wnt beta-catenin signaling, did not differ between both cell lines, indicating that LEF1 probably functions in a Wnt beta-catenin independent fashion, as it has also been recently described. So what we did next was to overexpress LEF1 in the less aggressive 410.4. And although we had overexpressed this EMT transcription factor, we didn't observe any induction of mesenchymal features, neither in the protein expression nor in the morphology of the 410.4 cells. But the overexpression of LEF1 led to a significant reduction of the migration and also the invasion capacity of these cells. And more importantly, it also improved their colonization capacity. Here you can see the left one overexpressing cells compared to the control 410.4 cells. So next, we had a look at the brain tissue, and we performed RNA analysis, in this case, real-time PCR. What we can see is that we can still detect the expression of LEV1 in the brain, but we see again no differences in axon 2. And more importantly, here in the tumor bulk, even though we had seen an improvement of the colonization capacity, we didn't observe any signs of EMT induction, neither here inside the tumor nor at the infiltration front. In both cases, in the control cells and also in the LEV1 overexpressing cells, the brain metastasis expressed high levels of the epithelial marker ikaderin at the infiltration front, which actually speaks against an EMT induction at the MMPI. So, okay, our initial hypothesis was that EMT induction might be responsible for the better colonization potential of the 41 cells in the brain. However, our data indicated that although LEV1 actually increased the colonization capacity of breast cancer cells, this was not mediated by an EMT induction at the MMPI, as it's usually the case in primary tumors. So we want to know how LEV1 contributes to metastatic colonization. And to answer this question, we performed a proteomic analysis in which we compare on the one hand the two original models, the 410.4 and the highly aggressive 41, and on the other hand, the 410.4 control cells with those with a LEV1 overexpression. In the first comparisons, we identified 122 differential expressed proteins, and in the second one, 65. And from this, 11 proteins were shared by both comparisons. So, of course, we had a closer look at these common differentially expressed proteins, and we observed that a high majority of them were related with glutathione metabolism. And as you know, glutathione is the main cellular redox buffer and protects the cell from ROS damage. So we hypothesized that the better colonization potential might correlate with the glutathione-dependent detoxification capacity of the tumor cells. And what we did first was we confirmed the expression of these targets in the cell lines with a better colonization potential, namely the 410.4 with the LEV1 overexpression and the 41 breast cancer cell lines. Next, we challenged the cell lines with the cellular ROS inducer TBHP. And as you see here, only the cell lines with a better colonization potential, the 41 and the 410.4 LEV1, only these cells were able to keep their intracellular ROS levels constant over time, while the control 410.4 cells enormously increased their intracellular ROS levels after addition of TBHP. Okay, so finally, what we did was we inhibit glutathione with BSO and check the ratio of oxidized versus reduced glutathione over time. And as you can see here, 6 hours after BSO removal, all cell lines decreased the ratios as expected. And interestingly, after 24 hours, the cell lines with a higher colonization potential, the 41 and the 410.4 with LEV1 overexpression, already recovered or restored their glutathione levels, while the control cells, the 410.4, didn't. So this could also be seen in a colony formation assay. Under BSO treatment, the number of colonies formed by the 410.4 control cell lines was significantly reduced, while the highly colonizing cells were not affected at all. I don't know if you're seeing my screen, but in my case, it stopped.
I think I see LEF1 modulates GSH metabolism slide now.
Okay, thank you so much. I want
You are
to
welcome.
the conclusions, but I think we have to wait for a second. Oh, I think
Now we see your desktop.
now it's gone. Okay, so give me a second, please. Can you see? Oh, I'm sorry. Can you see it now?
Yes, we can see it now.
But we're at the end. Okay, I'm sorry for that. I will go to the conclusions. Okay. So. Okay. So. Okay, so now I would like to conclude. Today I've shown you that brain metastases grow either by displacing or infiltrating the adjacent tissue, that metastatic infiltration correlates with a worse prognosis. There is at least 3 different MMPIs, displacing epithelial infiltrative or diffuse infiltrative, and we think that MMPIs are ruled by different molecular mechanisms, like I have shown you today in the case of strand and cohort infiltration with breast cancer models. And on the other hand, I hope I could convince you of the differences between the invasion front in the primary tumor and the metastatic infiltration front. Like, for example, in the case of our breast cancer cells with an epithelial MMPIs, whose colonization capacity in vivo depends more on their glutathione-mediated detoxification ability than on the induction of EMT. So, okay, but now I want to thank all the people who contributed to this work, especially my mentor, Tobias Bukrop, and my lab members, and also Martin Preswold from the Department of Neurosurgery, who provided us with the human samples. And I also want to thank you for your attention today, and I hope to meet you next time, maybe in person, but for now, you can also contact me at any time via email, and now I'm very happy to take your questions.
Thank you very much, Raquel. Very nice and interesting talk. I would like to ask you, who do you think that is the major contributor of brain metastasis, EMT or GSH metabolism? What would you say?
Yeah, so our results point to not only glutathione, but the metabolism of cells, how fit the cells are in the brain, because EMT, we thought we would find this EMT process at the infiltration front, but also in human metastasis, we didn't see any EMT induction at all. And we analyzed quite a lot of samples. So it seems that when cells arrive at the target organs, they really have to adapt to the new conditions. So that's why this is so important for them to be fit there. And for now, it's the question which differences we have in this fitness of the cells, and how can we target the different MMPIs so we can adapt the treatment of the patients to their MMPIs. This is what we hope to do in the next couple of years, and maybe I can present you more data next time. And
okay, thank you. And another question in relation with the infiltration. You show that was displacing an infiltrative kind of metastasis. Regarding the displacing, there is any difference in the overall survival of patients with the presence of the multi-layer capsule or not?
We didn't perform a big study with that. The only study we have done until now included only 39 patients, so we didn't have enough patients to have a look between all the different groups. But at the moment, we are working with the neurosurgery department, and we are having the samples from this area, from this MMPI, and we hope to have a bigger cohort in order to analyze all the different MMPIs. We see the MMPIs, but we still need to collect more patients in order to have more data about survival and have a bigger cohort of patients.
Okay, thank you very much.
Bye.